And so our approach is to simply approach HIV prevention with long-acting agents. And they could be in the form of potent and broad antibodies against the virus, or drugs that have been formulated to release very slowly and therefore last in the system for a long period of time. So let me begin by describing our more recent work on engineering antibodies. So more than half of my lab have become engineers, but engineers at the molecular level. And we're modifying antibodies to have greater and greater activity against HIV so that these antibodies could be administered in a passive way, given to people to substitute for vaccines. So a little bit of technical aspect first. Here HIV is on the bottom, the glycoprotein is shown here, and the receptor on the cell surface are first CD4 here and something called CCR5. So we know for HIV to penetrate into the cell, this needs to engage the primary receptor, this region here needs to bind to this region, and then this will twist and turn, engage the co-receptor, and the two membranes are then approximated and fused. And the internal contents here are injected into the cytoplasm of the cell. And in fact, there are antibodies that are not directed to the virus, but directed to the primary and secondary receptor. One molecule that we've been working on for a long time is an anti-CD4 antibody called ibelisumab. Another one is an anti-CCR5 antibody called Pro-140. And these were made initially generated in mice, humanized, and then given to patients, and found to be active in that indeed in patients, each of these antibodies could block infection and would lower the amount of virus in the patients, and have also been found to be safe, that is, no side effects in a number of human subjects. So I'm going to tell you about work that's done using this antibody and this antibody, as well as antibodies directed to the spike. And those antibodies are the same ones that I have pointed to in a previous slide. And I would just draw your attention to one called 10E8, which actually binds to the membrane proximal region of this transmembrane protein, which we call GP41. And we have created probably close to 200 engineered antibodies in the lab. I'm just going to tell you about one type of antibody that's particularly promising. Oh, before I get to that, the profile of these antibodies, as well as these two antibodies, in terms of blocking HIV infection, is shown here. And this is, I'm just going to walk you through this a little bit because many subsequent slides will be presented in this same format. So this is percent of virus coverage. And this is the antibody concentration. And in all these graphs, we're testing 118 strains of HIV. So it's a large library of HIV to their neutralization by these antibodies. So each line represents one antibody. And obviously, this is increasing concentration on log scale. So the more potent the antibody is, the profile should shift to the left because you need less of the antibody to neutralize some or most of the viruses. And you could see, for example, the antibody to CD4 or CCR5 to the receptors, the CD4 one is here, the CCR5 one is here in purple, and then these are directed to various parts of the viral envelope glycoprotein, and they have profiles like these. So some are way shifted to the right, that means they're not very potent, and some don't reach the top, that means they don't get all the viruses. They get maybe up to, for most of these, 80–90%. One of this is actually broader. And then we said all of these are not good enough, we think. And in fact, when you mix 5 of these antibodies together, you get this black profile, which now covers all the viruses and with the potency shifted to the left. Okay, so gain in potency. So, but 5 antibodies to give is a mess. It's too expensive and so on and so forth. So, but this becomes our benchmark. We want to do better than that. So what we've been doing, and when I say we, it's actually 2 colleagues in the lab, Jian Yu and Huang Yaoxing, two rather senior colleagues in the lab. And I'm going to present only data that they have generated on this type of antibody that we have engineered. This is, they are bispecific in that one arm, antibodies usually, both arms grab the same thing. But in this case, one arm grabs something and the other arm grabs something else. And they don't naturally occur that way, so we have to engineer them. So so we would take the heavy chain gene of one antibody and the light chain gene of another antibody and mix it in with the heavy chain of a second antibody with the light chain of another. But for those of you who think about these things, if you mix those 4 genes in, you're going to get 10 different antibody species because the pairing will be more or less random. So how do you make sure the pairing is of the specific type you desire? Well, we want the formation of 2 different heavy chains coming together. We don't want the homodimers, we want the heterodimers. And so one could create a hole and a knob so that you would discourage the pairing of homodimers and favor the pairing of heterodimers. And indeed, that is the case when you do so. But the light chain could mispair. So this light chain could mispair with this heavy chain, and then that's no good. So on the one side, you don't touch it, so this pairing would occur naturally. But the other side, as a trick that actually was developed by scientists at Genentech, is you cross over CH and Cl. So when you cross it over like this, you would still have proper pairing, but this region would look the same and would bind to the epitope just the same. But now this light chain will not mispair with that heavy chain. And when you do that, I won't go into the data, but when you do that and characterize it by native mass spec, so you just fly the entire collection of antibodies in your soup into the mass spec without chopping it, without processing the protein. And this native mass spec has a resolution of 1 Dalton for 150 kilodalton protein. And you could see that we get essentially, these are different charge forms, but if we just look at this one, this state in red, essentially 87% of the antibodies that we have in that preparation is exactly what we want, the bispecific form. And then the other forms are rather minor. And so this is pretty reassuring. And then we have constructed this library of antibodies with the left arm binding to various components on the envelope glycoprotein, and on the right arm binding to one or the other cellular receptor. And I would tell you that about 95% of the bispecific antibodies confer no benefits, in that we don't have greater potency, we don't have greater breadth. And those results are demonstrated on 3 panels here. So if you look at one parental antibody or the other parental antibody or the bispecific antibody in terms of their potency, the lines are every which way, right, in these 3 panels, as if the bispecificity offers no added advantage. But the distinction is here. You see one antibody here, another parental antibody here, and then the lines typically go down like a V, suggesting that there's greater potency for the bispecific. And in fact, we have, from that library of 100-something, 2 stood out for us. And both of them are shown here in red and in blue. And you could see, compared to the mix of 5 antibodies now in that black line that I highlighted earlier, both are approximately the same or superior. And this dark blue line is in fact 10E8 and anti-CD4 we call IMAP here. Again, the technical details are not important. So if one arm is grabbing the receptor here, and the other arm is grabbing the membrane proximal region of the virus protein, it's neutralizing so well. And then what was actually shocking to us was 10E8 Pro-140. So again, it's the 10E8 and the Pro-140. Pro-140 now is grabbing the co-receptor rather than the primary receptor. And the potency is about a log better. We have done a lot of studies to address synergy and mechanism. I don't have time to go into them, but the synergy is pretty obvious in that look at this line, for example, 10E8 Pro-140. What are the profiles of the parental antibodies? And they are here. Pro-140 is this dotted red, 10E8 is this dotted green. So they're 2 of the weakest antibodies, and somehow when they are joined together, we get this activity. When you mix these 2, just mixing them, you will have something over here, not over here. So it's not just the 2 active agents working in concert in synergy, but the physical linkage of those 2 elements is required. And we believe this is largely working by the mechanism of concentrating 10E8 on the cell surface at the right place at the right time. So it's location, location, location. It's concentrating the active agent at the very place the virus is trying to penetrate into the cell. Now, when we are developing this as a prevention method, so you have to get down to practical issues. And so it's part of product development. It's antibody engineering and product development. And luck would have it that while we get extreme potency and breadth, we have problems. We have stability problems. And to make a long story short, it seems that there's something unstable about both of these antibodies. So when we run the protein through size exclusion chromatography, you could see these 2 antibodies that we like both have double peaks, suggesting there are 2 species, which we're still trying to understand. When you make another bispecific antibody without the 10E8 component, you see a nice single peak. And you go back to the parental antibodies, 10E8. So we know it's coming from the 10E8 side. There's something about that antibody gene sequence that is causing stability problems. And worse yet, that stability problem is not just lowering the amount of your product, it also gives you very bad pharmacokinetic results in animals. So it's clear or lost in the body for whatever reason. So fundamentally, it's not a product until you solve this technical issue. And we try to solve this problem. Again, it's mostly Drs. Huang and Yu. There are production conditions you could modify to see if that would be, and we tried many without success. There are formulation conditions, buffer conditions. We tried many, again, no success. There are published solutions to a lambda light chain problem, and this is a lambda light chain. Again, no success. So we had to go back and say, okay, so we know it's coming from the left side of that bispecific antibody. And the antibody has evolved a great deal. I told you that it took years of evolution to get to that antibody. So you could say, then for that antibody, we know what the evolutionary path was. Luckily, those studies were performed by scientists at the NIH. And so we know there are lots of sort of ancestral variants of the 10E8 that exist. So we could say, can we take those variants, which we call somatic variants, and see whether any of those would substitute? And in fact, there is a somatic variant which we call variant 1.0 that gives a nice single profile on this size exclusion chromatography. And when that variant is put into mice, it gives a nice pharmacokinetic profile. But the problem is that this variant is not very potent. So we have a very potent form that's not stable, and then we have stable forms that's not very potent. But these sequences are highly related. So it gives us hope that if you now start to play with the genetics, you make mutations, you make hybrids, you might find one that would become stable and retaining the potency. And in fact, that's what we found after creating about 40 different variants. And here I'm just schematically showing the variations in the heavy chain gene and the light chain gene by these dots. And you could see that variant 1.1 here, with these modifications here compared to the original sequence, so this took a lot of work, has great activity, has great stability based on the size exclusion chromatography, and great pharmacokinetics. And similarly, for another species that's based on iberalizumab rather than Pro-140, you see this variant 2.0 is also very good. But interestingly, this variant 2.0 in this context is no good. And interestingly, variant 1.1 in this context is okay, but we don't have the complete data. So it's not only certain sequences, it's the context of those sequences. So this has been a puzzle-solving exercise to figure out what might do all this. But nevertheless, these now are two molecules that we like in that they have great activity and stability. So the activity profile shown here for this variant 1.1, Pro-140 is this profile here. Now compared to that 5 antibody mix in this dotted line, it's good. And then this variant here, and you see their profiles indicating stability are good. So in the end, we are now looking at these two antibodies to consider them for further clinical development. The rest of the field, in fact, are developing these antibodies. And previously, I didn't talk about it. Previously, we took this antibody and made it better. And this antibody is actually in production to get into a phase 1 study. And that's a glycan-modified form of iberalizumab. So our group has come up with these 3 antibodies, all of which are now candidate for what we call passive immunization for HIV prevention. But all 3 of them are in the preclinical phase, where some of these have entered the clinic already. So that's the first subject. Let me shift gear and talk to you about something very different, and something that is in fact further along in this HIV prevention world.